What's good, everyone? My name is Kashi. In today's episode, we're going to be discovering a new type of G protein called the GQG protein. And we're going to take a look at what enzyme the GQ protein activates and what the responses of this enzyme are. Let's get into it. So, for this video, we're going to follow a pretty similar format as we did for our last video. So, in our last video, we looked at the new types of G proteins, GS and GI proteins. And we looked at what these G proteins do, which is activate the effector enzyme adenyl cyclase. And we looked at what adenyl cyclase does, which is produce the second messenger, CEMP. And we looked at what second messenger CEMP does, which is activate protein kinases to produce a response. So, using the same format, let's now apply it to our new example. So to start off, our new type of G protein that we're going to focus on is called the GQ protein. Let's take a look at how this G protein gets activated. So if you guys are looking for a fully comprehensive review on G proteins, how they work, and how they're produced and terminated, be sure to check out my video entirely dedicated to G proteins. I'll link it up at the top. But in today's video, we're just going to really briefly go over how GQ proteins are activated. So like with most G proteins, you got your cell membrane, and you have your GPCR receptor, which is bound to it. And on the inside of your cell, you have your G protein bound to your GPCR, and your four components. So you have alpha, beta, gamma, and you have GDP because it's inactive right now. And this G protein specifically is of the GQ type. So lots of different types of G proteins. This one is a GQ protein. So a messenger molecule comes along, right? And it binds to your GPCR receptor, and now your GDP leaves and it gets replaced with GTP. So we can replace this GDP with GTP, and now your GQ protein is active. So it can split into its two different components, your GTP alpha components and your beta gamma components. And they can go off and activate different enzymes. And your GTP alpha components can go off and activate the enzyme known as phospholipase C. Alright, so returning to our flowchart, we've outlined that our G protein is known as GQ, and what it does is it goes and activates the effector enzyme called phospholipase C. Phospholipase C. So now, let's take a look at what phospholipase C does. Alright, so before we can get into what phospholipase C does, we first need to review the structure of the cell membrane. So here, you have a cell, and here is a diagram of the cell membrane. So as you can see, here we have the outside of the cell, and here we have the intracellular part, the part inside the cell. And in between, we have the cell membrane that separates the inside of the cell from the outside of the cell. And as you can see, the cell membrane is a bit complicated. It's made of these two layers of things. You have this top layer up here, and you have this bottom layer down here. Well, what are these layers? Well, actually, the cell membrane is what we call a phospholipid bilayer. Phospholipid bilayer. So bilayer means layer of two. So there's two layers, right? Two layers of phospholipids. What are phospholipids though? Well, you see each of these things we have here in our cell membrane, each of these little components with the yellow circle and these two red lines, each of these represents a phospholipid. So let's zoom in and see what this is. A phospholipid, basically it consists of, so you have the head, right? This is the head, which has a phosphate group bound to a glycerol. The phosphate group is where it gets the name phospho. And it gets the name lipid because this bottom half, here you have two fatty acid chains. So, just to cover that again, each of these phospholipids contains a head with a phosphate group attached to a glycerol, and then down here you have your two fatty acid chains. So the part down here is fatty, meaning it is nonpolar, and nonpolar things don't like water, so this is hydrophobic. However, your phosphate group and your glycerol up here make this part at the head, make the head polar. Therefore, this likes water, so it's hydrophilic. So as a result, the head of your phospholipid is polar and it likes water. The tails of your phospholipids are nonpolar and they don't like water. And that's why the phospholipids have arranged themselves like this. Because if you think about it, inside the cell, you have water you have like liquid in the form of cytoplasm, which contains water. So as a result, I'll put a little blue asterisk to denote that it's water, watery, wet, whatever. And on the outside of the cell, you also have different fluids, liquids, blood. So I'll do a little asterisk there to show that it's wet. So that's why the phospholipids have arranged themselves like this. As you can see, the polar, like the head of the phospholipid, which is the polar hydrophilic component, is pointed towards the outside of the cell and the inside of the cell, because those are the parts that are attracted to liquid because they're polar. And the fatty acid chains, which is the nonpolar components, they don't like water, so they want to stay away from water. And as a result, they are found on the inside of the cell membrane, far away from water. And that's why the phospholipid bilayer is arranged like this, with the polar heads on the outsides towards water and the nonpolar tails on the insides. Another thing to note with phospholipids is that they're not all the same, and there's actually different types of phospholipids. So let's say this one right here, right? So the phospholipid type that we're going to focus on is, well, here's the full name. It's really annoying. I'm not going to try to say it. But the short form for this is PIP2. This is the name of your phospholipid. And this phospholipid is significant because this is what our Phospholipase C enzyme acts upon. Let's figure out how this works. Okay, so once your messenger molecule binds to your GPCR and your GQ protein gets activated and your GTP and alpha components, they can make their way over to your phospholipase C and activate this effector enzyme. And as you can see, the phospholipase C enzyme is embedded in your cell membrane. And remember, your cell membrane is made of a phospholipid bilayer. So you have all of these phospholipids and two layers of them. And actually, this also continues here on this side. I just haven't drawn it because I'm lazy. But you have your phospholipid bilayer here. And what your phospholipase C does is it cleaves PIP2. Remember, PIP2 is a specific type of phospholipid. So let's say, we zoom in, let's say this phospholipid right here is PIP2, right? So your phospholipase C, what it does is this enzyme, it goes and it cleaves this phospholipid. And when I say cleave, I mean it splits it apart. So let's say it splits it down the middle. It splits apart your nonpolar components and your polar components. And what do we know about your polar component, which is your yellow head? Your polar component likes water. So this goes off into your cytoplasm where there's liquid. So that gets removed from I did not mean to do all that. So that gets removed from your cell membrane, and what's left is here, your nonpolar component, which doesn't like water, so it stays here tucked away in the cell membrane. So this component, which is polar and goes into your cytoplasm, is known as IP3, or inositol triphosphate. Inositol triphosphate. And your Your nonpolar component, which remains in your cell membrane over here, is known as DAG, or diacylglycerol. So basically what your phospholipase C does is it cleaves your PIP2. It splits it into IP3 and DAG. And your DAG does not like water, so it's nonpolar, and it stays in the cell membrane. Whereas on the other hand, your IP3 is hydrophilic, so it's polar, it likes water, and it goes into the cytoplasm. So just to recap that one more time, you have your phospholipid PIP2 in your cell membrane, and phospholipase C comes along and splits it, so you have one polar component in your cytoplasm and one nonpolar component remaining in your cell membrane, IP3 and DAG. In fact, IP3 and DAG are actually your second messengers. So let's go back to our flowchart and make this clear. Alright, so now we've established that when our GQ protein activates our enzyme phospholipase C, what phospholipase C does is it cleaves our phospholipid in the cell membrane called PIP2, splitting it into two components, IP3 and DAG. And these are our two second messengers. So let's write them over here. You got IP3 plus DAG. DAG. And IP3 is your polar component that goes into your cytoplasm inside the cell, whereas DAG is nonpolar, so it remains in your cell membrane. So now that you've made your two second messengers, just like CAMP, these different second messengers can go and produce responses. Let's take a look at this. Alright, so now we've got our two second messengers, right? IP3 and DAG. So your DAG second messenger, that's the one that stays in your cell membrane because it's nonpolar. And what this second messenger does is it activates a kinase called kinase C. Now, if you want to learn more about kinases, in our last episode about adenyl cyclase and cAMP, we looked really in depth about kinases, so if you want to learn more, just be sure to check out that one. I'll link it up at the top. But kinase C is basically a specific type of kinase, and like most kinases, it's good at phosphorylating things, so it can add a phosphate group to enzymes or other substrates, so it phosphorylates them and adds an enzyme. And in doing so, it can lead to various different responses. So that's our first second messenger out of the way. DAG, we've got that it activates protein kinase C. Now let's take a look at IP3. So unlike DAG, IP3 doesn't stay in the membrane, but it is instead found inside the cell in the cytoplasm because it's polar. And what it does is basically inside the cell, right? You have different storage compartments for various different things, such as vesicles or something called the sarcoplasmic reticulum. And what IP3 does when it goes into the cytoplasm, it stimulates the release of calcium from these intracellular stores. So calcium is released into the cell's cytoplasm. And this calcium can do different responses. For example, calcium is essential for smooth muscle contraction. So basically, this IP3 can cause calcium to be released from storage components inside the cell, and this can cause the contraction of various smooth muscles, such as vascular smooth muscle or other types. So now we can finally finish up our flowchart. So we've established our two second messengers, and these second messengers can do the response of the following. So IP3, that's the one in our cytoplasm, that does the response of calcium release, calcium ion release from intracellular stores, which can do things such as stimulate muscle contraction. And then DAG, which is the second messenger that stays in the cell membrane, can activate protein kinase C, which can lead to more cell responses. So now you can see the contrast between these two different systems. In the first system, our G protein are the GS and GI proteins, which control adenyl cyclase, produce the second messenger CAMP, and activate various kinases to produce response. Whereas here, very similar, except we have a different type of G protein called the GQ protein that activates phospholipase C instead of adenyl cyclase, which cleaves PIP2 to synthesize our two secondary messengers instead of CAMP, and then each of these can produce their own responses. Another important thing to note is that just like any type of G protein, our GQG proteins are terminated by enzyme GTPase. This enzyme dephosphorylates our active GTP into GDP by removing a phosphate group, and by this it turns our active GQ protein into its inactive form so that it can combine again with the beta and gamma subunits and return to the GPCR. And this applies to any type of G protein, whether it's GS, GI, GQ, or any others. And this is important because remember, looking again at our cell signaling framework, after response must always come termination. Otherwise, our response will just go out of control, and this can be very dangerous. So, to conclude, I hope that this video was able to give you guys a greater insight of this new type of G protein called the GQ protein, the enzyme that it activates, the two second messengers that it produces, and the different responses that they produce, as well as a way that this G protein can get terminated by GTPase. I hope this video was helpful and easy to understand. If you guys have any tips or suggestions or comments, please let me know down in the comments below. I'd really appreciate it. And in the next episode, we're going to start applying our cell signaling framework to some specific messenger molecules, such as maybe histamine or acetylcholine or catecholamines. I'll decide and we'll see in the next video. But for now, thank you so much for watching. If you liked the video, please consider liking and subscribing, and I'll see you guys next time. Take care.